Hey guys, I'm going to go over your macromolecule slideshow. So today everybody should be working on the macromolecule sheet. You'll watch this Amoeba Sisters video. You will be completing this sheet as well. Follow the directions on that. Let's go over what we have here. First, let me get my little self out of the way. More prefixes. And let's science count. Okay, science count. 1 is mono, 2 is di, 3 tri, 4 tetra, 5 penta, 6 hexa, hepta, octa, non, dec. Let's try that with no. Okay. 1 is mono, di, tri, tetra, penta, hexa, hepta, octa, non, dec. That's just a good thing. Poly means many. Philic is loving. Phobic is hating fear. So think about a term hydrophobic. What would that mean? And that is fear of water. Hydrophilic likes water. Good. Okay. Organic compounds means contains carbon. Organic means to contain carbon. Inorganic means does not contain carbon. All living things contain carbon, which forms really long chains. Macromolecules. Macro means large. So these are the large molecules. A monomer is a single unit and a polymer is many units. There are 4 major macromolecules that are basis for life, and macro is big again. So, and they all are composed of smaller single units, which is monomer. Mono is 1, poly is many. The formula of glucose, and this is one of our first. This is actually a carbohydrate. This fits into carbohydrates. When we think of carbohydrates, we think of quick term energy. The formula for glucose, and I'm going to have you guys sing it, is C6H12O6. I'm going to say, what's glucose? You guys are going to say C6H12O6. C6H12O6. C6H12O6 is the answer. C6H12O6, that's glucose. It's 6 carbons, 12 hydrogens, and 6 oxygens. It is a ring-like structure. You can see that here. It comes from the foods that we eat. The foods that we eat. Cells in our body take in glucose and use it as energy. When we eat our chemical potential energy, so our breakfast, okay, in the morning, that is the food, and we get glucose from that. From eating our food and taking in that glucose, we are able to do something called cellular respiration, where we take in oxygen and glucose and we give off carbon dioxide and water. The 4 macromolecules are proteins, carbs, lipids, and nucleic acids. We're going to briefly discuss all those. We just had a little discussion just now over one carbohydrate, which is glucose. Carbohydrates, they're made up of carbon, hydrogen, oxygen. The monomer of carbohydrates are monosaccharides, mono being 1. An example of a monosaccharide is glucose. They give us short-term energy or quick-term energy. If I have to run a race tomorrow, I'm gonna carb load tonight. If I don't burn those carbs, though, this is the problem. What happens to carbs? They get stored as fat, as lipids. So short-term energy, you eat a lot of sugar, you eat a lot of sweets. What happens to us? We go, oh, we don't feel good later. We have to burn off those carbs. So carbohydrates, the monomer, monosaccharides. Carbohydrates and sugars typically end in ose. So fructose, galactose, lactose. Look at the back of your water bottle, or not water bottle, I'm sorry, your soda. It's going to say probably high fructose corn syrup. That's sugar. Cellulose, that's found in the cell walls of plants, and it's a carbohydrate. Look at it. It ends in ose, O-S-E. It's found in the cell walls of plants. It's very difficult for us to digest. Now, here's another example of a carb. It is found in chitin. It looks like chitin, but it's chitin. It's in the exoskeleton of arthropods. So chitin. So these are carbohydrates. Lipids, carbon, hydrogen, oxygen, they have a different ratio. They actually do not have a monomer. Lipids don't have a monomer, but they're made of fatty acids and glycerols. They give us long-term energy. This is adipose tissue. So long-term energy. Examples, fats, oils, waxes. And think about this. Are these polar or non-polar? So remember the other day we learned about being polar. Polar is hydrophilic. Polar is is unequal sharing of electrons. Water is polar, so it's hydrophilic. Nonpolar is unequal sharing of electrons. So these are nonpolar. They don't mix with water. We've also, I think briefly, I don't know if we went into saturated and unsaturated fat. So just a good tip for you, a type of fat, good fats are unsaturated and bad fats are saturated. So bad fats are like bacon grease, Crisco, like the hard stuff, and oils like olive oil, Wesson oil, vegetable oil, those are good oils. And we typically look at the bonding in those. All single bonds, all single bonds are saturated fats. If it has at least one double or one triple bond, it's unsaturated. And that's something I'll have to show you in class. Lipids found in the cell membrane. They're nonpolar, hydrophobic, they're water-fearing. So look here, and this is a question you may have on ESC. Where might you find lipids in every single part of the body? And it's going to be the cell membrane. Every organism and all cells have a cell membrane, or also called a plasma membrane, or it's I'll leave it there. Okay. This is the phospholipid bilayer. These tops right here, these are the polar heads. These are the non-polar tails. It's a double lipid bilayer. This is the double part. So there's two of these little sections right here. This part is polar. This part's non-polar. So this would also be hydrophilic. And this is hydrophilic. And these two, since they're non-polar, they would be, what's the word? Hydrophobic. Lipids found in fat. Here we go. Single bonds, very good right here. Saturated, these are the bad ones. If they're solid at room temperature, this is also what they do in your arteries. This is what they do. If you cook that bacon and leave that grease there and then come back later and it's hard in the pan, that's saturated fat. That's the bad fat. Olive oil, healthy oils, these are unsaturated. They have at least one double or triple bond. Here we go. All right, I want to go over why can't Miss M eat ice cream? Have you ever heard of anyone who's lactose intolerant? Well, lactose means sugar. That's the sugar found in milk. Well, we all need enzymes to break that down. If your enzyme lactase doesn't function properly, you can't break that down. So here's a hint, and we will get there. Enzymes typically end in ACE. So lactose, that's a carbohydrate, and that's a sugar, and it ends in ose. Enzymes end in ACE. So we're talking about proteins with the lactase. Carbon, hydrogen, oxygen, nitrogen. Here's something else. So they contain nitrogen. Monomer, or the building blocks of proteins are amino acids. I'm going to ask you this. How many amino acids are there? There are 20 different amino acids. Proteins, this is the highlighted one. It's involved in every biological function in the body. If our proteins don't function, we don't function correctly. Proteins control everything. Proteins are also, you're going to hear the word catalyst. What do? They speed up a reaction. How do they do that? By lowering the activation energy. So proteins are made up of amino acids. How many are there? 20. Give me an example of a protein. Lactase. Insulin. We know diabetics use that. Our body naturally makes that. It's found in muscle fibers, enzymes, antibodies. But these are proteins. They control every metabolic function. They are enzymes, meaning they are catalysts. And this is what I just said. They lower the activation energy. So here is the reaction. This is the reaction without a catalyst, and this is with a catalyst. The reaction will do everything the same, except this part, it doesn't need as much energy, so it lowers the activation energy. Lactase is an enzyme that helps us break down milk sugar. So let me give you an example of this. Have you guys ever been to Six Flags and maybe rode Goliath? It's a really, really, really big coaster. It's the longest coaster at Six Flags. Because it's so long, it has to have that really, really, really high hill. So it would, at that first hill, it would actually be nice just to ride Goliath without having to do that really scary first hill. But so if we had Goliath without or with an enzyme or with a catalyst, it would lower that first hill. But if we lower that first hill, it's not going to be as long. So how do catalysts speed up reactions? By lowering the activation energy. The energy needed to start the reaction. But everything else after that is going to be the same. Okay. Move myself over here again. All right, nucleic acids. These are made up of carbon, hydrogen, oxygen, nitrogen, and phosphate. The monomer are nucleotides. What is a nucleotide? A sugar, phosphate, and a base. What's a nucleotide? A sugar, phosphate, and a base. I'm going to have you even sing it. Sugar, phosphate, and a base. These are the monomers of nucleotides. They're found in all living things. It's the information storage molecule. It is our genetic material. One example of a nucleic acid is ATP. It carries the energy carrying molecule. ATP, which is adenosine triphosphate, can be converted to ADP, which is adenosine diphosphate. What does di mean? 2. What is triphosphate? 3 phosphates. When this reaction occurs and that phosphate is broken from here, energy is released. That's where we get energy. But that's one nucleic acid. The others that we also think of are DNA and RNA. The sugar in DNA is deoxyribose. RNA, ribose. The phosphate group is the same for both. The bases in DNA at Greene County, ATGC. It's actually adenine, thymine, guanine, cytosine. RNA is AUGC, so it is adenine, uracil, guanine, cytosine. The shape of DNA is double helix, and this one is a single helix. These are nucleic acids. Chargaff's rule, love, love, love this one. All right, Chargaff's rule. This, Chargaff is a scientist. This just shows you how the pairing goes in DNA and RNA. When DNA makes a copy of itself, A always pairs with T, G pairs with C. It's a simple rule, okay? Let me explain this one. A always pairs with T, G pairs with C. So here's the math on this, and you will have this question. If we have 10% adenine, how much thymine would we have? Well, if you have 10% adenine, how much thymine? 10%. Okay, what's 10 plus 10? 20. How much of guanine and cytosine would you have? I'm going to give you a second to think about it. It's going to be in equal portions. So if that's 10, that's 10, that's 20. 100 minus 20 is 80. 80 divided by 2 is, you guessed it, 40. So that's 40 and 40. And here's another thing. We have purines and pyrimidines. The purines are adenine and guanine. The pyrimidines are thymine, cytosine, and uracil. The way I remember this is purines is the short word, and there's only 2 of them. And pyrimidines, there are 3 of them, and it's the long word. Thymine, cytosine, and uracil. And that is your last. We'll do your last little review, and then we're done. What foods are made of carbohydrates? I'll give you a second. Sugars. What's the monomer of a carbohydrate? Monosaccharide. What's a monosaccharide? Let's say monosaccharide. Disaccharide. What function do lipids, fats serve for seals? What do they do? Reserve. What types of foods are high in protein? Meats, nuts. Proteins are made of what monomer? Amino acid. How many amino acids are there? 20. What is the monomer of nucleic acids? A what, a what, and a what? A sugar, phosphate, and a base. What two molecules are made of nucleic acids? We named three, name two. We named ATP and then DNA and RNA. And that's it. Thank you. Stop.